and share the screen.
Dear all, thank you for coming here. I know how difficult this period is for all of you. Dear Vasilis, thanks for introducing me. During my early years in my master thesis, I studied emotion-aware computing. We used emotional stimuli, visual emotional stimuli, selected from the International Affective Picture System in order to investigate how the brain responds to various emotions. As you may know, the current emotional theory is based on two orthogonal variables. The one is the arousal dimension, how much excited I am. So if, for example, a lion is coming across me, I am very aroused. But I am also, I could also be happily aroused. Whether the emotion is is a positive one, happy, or a negative, this is the second dimension, which is the valence. Both dimensions are vital for our survival and for our evolution. So we used these stimuli in order to record event-related potentials. What event-related potential means? As you know, the brain has its ongoing activity for our entire life. But when I see a stimulus, for example, the lion coming across to me, this, I should react to this. It is the fight or flight response. So there is a response that is superimposed to the ongoing EEG activity. However, this superimposed signal is a relatively small one regarding to the random noise of EEG activity, and it may be diminished. If we average same stimuli, then I get a very clear signal, a signal with high signal to noise ratio, and I can detect how strong my response is and how fast it is. We found that that there are gender differences in those responses. Males respond either with the left or with the right hemisphere, but on the other hand, females tend to activate both hemispheres simultaneously. This was a study published in Brain Topography and was among the best and the early gender differences studies. Then I used multimodal signals from those event-related responses, electrodermal activity, cardiac responses, oculomographic activity, in order to build some emotion classifiers. During my first study, these classifiers were based either to distance-based classifiers, such as or decision trees, and it yielded an accuracy around 78%. Later on, I also employed support vector machines, and I tried to integrate this gender information in order to build two separate classifiers, one model for males and another model for females. And this yielded to significantly better results around 84%. The studies were published in IEEE Transactions on Information in Biomedicine at 2010. Then we applied this know-how to a computerized neuroplasticity program. This program combined cognitive and physical training through computers to senior citizens in order to promote active aging and independent living. We also applied a computational scheme that used electroencephalographic activity in order to measure how the activity from one sensor is synchronized with the activity of another sensor. We used discrete wavelets, the orthogonal discrete wavelet transform, in order to estimate the cooperative activity in terms of spectral frequency. What we found is that when we represented this cooperative activity in terms of networks and we analyzed through graph theory, there were some correlates, some statistically significant correlations among the brain function and the performance in neuropsychological testing, in neurocognitive markers used for estimating the generic condition. These are the mini-mental state examination, which identifies whether a patient is demented or not, and also the Montreal Cognitive Assessment test, which also can discriminate the transitional states of mild cognitive impairment. And it was astonishing to find that both these behavioral examinations correlated with the notion of small world property. It is a concept which quantifies whether a network is efficient or not. If the network follows this small world property, it means that it could process the information very strongly on a local basis. For example, my visual cortex responds rapidly to the lion, but it also flows the information to other brain regions like the prefrontal cortex in order to take some action. Regarding the significance of specific nodes, we found that in healthy aging, there are some significant nodes either on anterior sides or on posterior sides. However, as you may see here, in the mild cognitive impairment case, these patients lose their capacity of their posterior hubs. And they built some new ones on right anterior sides, which were not activated in healthy aging. So the brain may lose its capacity, but it still fights to compensate by recruiting additional regions. And there is no obvious evidence of behavioral degradation on everyday living, or at least a significant one. However, later on the clinical onset of dementia, the brain loses its activity to compensate, and there is a clinical onset of neurodegeneration. We applied this brain metrics in order to build some images, some RGB images. These images describe the cooperative activity of the brain regions under several spectral scales, for example, under several frequencies. And then we built a convolutional neural network in order to predict the actual age of the participants regarding their brain response. And when we applied this training model to some patients, either from mild cognitive impairment or mild dementia, we found, of course, some deviations among their actual age and their predicted age. Maybe it is a failure of a good training, but happily no. This deviation of the CNN's output from the actual age was also correlated with the symptoms severity. And we actually could predict both physiological aging and pathological aging mechanisms with a very good accuracy. So, next, I participated in an experiment held at the premises of European Space Agency in Cologne, Germany, in cooperation with the Deutsches Zentrum für Luft- und Raum. This is the Envy Hub building at Cologne. You may see that all the rooms, all the facilities are below the ground. And it was built by German Aerospace Agency in order to resemble a space station. Within this Envy Hub building, there are 12 rooms like this. In each one room, a participant stayed there for 3 minutes. For 3 months, sorry. And for the 2 months, for the 8 weeks, he was confined to a bed with a head-down tilt. So you may see that the bed is 6 degrees lower than the head. And this causes the bodily fluids to shift towards the upper body case. And this head-down tilt bed study is the most accurate simulation of microgravity. So our aim was to study how the body responds to a long-term weightlessness. Bear in mind that these participants remained 24 hours per day for 8 weeks on that bed. What we wished to model is the situation that they will face either to the International Space Station or to a long-term Mars exploration. You know that isolation, confinement causes many problems in our mood, which may deteriorate our sleep quality, and actually it can affect our cognitive performance. Imagine, for example, that all of us will stay for in that room for a year. How does it look like? So, the experiment involved initially 14 days of baseline data collection. Then participants remained at this head-down tilt position for 60 days, and actually there were 7 to 14 days of recovery. And I tortured the participants by gathering entire night polysomnographic studies for 5 days, for 5 nights during that period. What we found is that during sleep, the brain is not inactive. Actually, there is a dynamic functional interplay among the various cortical regions during the various sleep stages, starting from non-REM 1 to REM sleep. And you may see how the cortical activity spans from posterior to anterior sides in very high speed. And this is repeated during the entire night. Also, the default mode network regions tend to organize into different sub-modules. So the brain explores different states in order to be able to respond to the challenges of the next day. This was our computational pipeline that we applied. We gathered the data. We applied time series techniques in order to get high-quality artifact-free data. Then we applied cortical modeling. We modeled the time series of various cortical regions in order to obtain functional connectivity analysis and finally to apply graph theory metrics. The first outcome of this study was an automated sleep staging employing convolutional neural networks on those default mode network activations. The problem was that we had 4 different stages, and non-REM 1, non-REM 2, non-REM 3, and REM, and we wished to have a model, a machine learning model that would apply automatic sleep staging. Otherwise, it is a tedious task for a sleep expert because for 8 hours of sleep recordings, the expert needs 2 to 3 hours to score those data, to identify which state is active. Also, another problem that falls within the domain of artificial intelligence bias is that the first transitional stage of non-REM 1 is only 2 to 5 minutes of the entire night. So the initial CNN model tends to overlook those data and to classify erroneously. So we applied the synthetic minority oversampling technique, this mode, in order to obtain surrogate data for that small minority cluster. What we obtained is some RGB images for each sleep stage. This is the RGB image for N1, this for N2, this for N3, and this for REM sleep. You may know that REM sleep is the stage where we are dreaming, and you may identify some similarities with the non-REM 1 sleep. It is the stage that we fall asleep, but we have some consciousness. It is important because actually the sleep data when we are dreaming look like an awake EEG activity. So this was the convolutional neural network approach. It is a relatively shallow network. And these are the regions that we recorded its activity. We left some data for testing, and thanks to that network architecture, we applied a very high accuracy, and this study was published at the IEEE Transactions on Neural Network and Learning Systems. It is a highly prestigious journal with impact factor above 14. So, you see here two pictures. The one is from the first spacewalk of the Team Peak, which is the first British astronaut that performed a spacewalk 2 to 3 years ago. On the right, there is a bedridden senior elderly. Do you observe any similarity among those two pictures, or think that they are very different? What do you think?
Life support systems.
So it is hardly to identify any similarity. On the other hand, they suffer from the same condition. It is lack of gravity. Gravity is our friend. We are healthy because we feel the gravity. And why is that happening? Because our muscles remain at a challenging position. If I have to walk half an hour or 2 hours in my everyday life, then my muscle system is activated and remain healthy. On the space, there is minimum gravity. So the first problem, not the first, because the first is the motion sickness, but the second problem that most of the astronauts suffer is muscle atrophy and cardiovascular atrophy. So it is the same as bedridden senior citizens. So what we aim to do is to find some methods to mitigate the detrimental microgravity effects. The most common one that is already applied to the International Space Station is ergometric bicycles or other ergometric devices for running and walking. And actually, they work well, very well, for both the cardiovascular and the muscular system. But the ultimate goal is to suggest a mitigation approach that could challenge all the physiological systems of our human body at once. And this system actually is there, but the problem is that it is relatively bulky in size. So it is the short-arm human centrifuge, either only with artificial gravity training or combining it with microgravity. And okay, you may see here that one person is exercising, me, and there is also a 90-year-old senior citizen that performs artificial gravity. Simultaneously, we can record both EEG activity and some biosignals like cardiac output, blood pressure, mean arterial pressure, etc. This is our infrastructure. Please bear in mind the infrastructure because there are collaboration opportunities. And we can perform multimodal data collection. The first research question is whether to answer whether artificial gravity training is actually an exercise. And we noticed that as the gravity load increases, the heart rate intensity also increases. For example, it is different if I just walk or sprint. If I run at a fast mode, my heart rate increases, and there is a formula that it is the maximum heart rate intensity for each one of us is 22 minus our age. So the closest we reach to that threshold, the more intense the exercise is. Please do not get away from that threshold because it is a life-threatening situation. And we may also see the cardiac output that also increased as the gravity load increased. And we observed some statistically significant results, both in terms of the cardiovascular system and also functional connectivity. We applied this method to a cohort of healthy young volunteers. This is the article published. And we also applied a similar protocol to some patients suffering either from multiple sclerosis or from stroke. And 2 months after the training intervention, we observed some functional connectivity changes in regions of the temporal cortex, which is plastic to physical activity training, and also to limbic or prefrontal cortex activations, which were also associated with a neuroplasticity effect. So the next stage is to apply artificial intelligence solutions in order to investigate which is the optimal dosage training for each one of us. Again, there are gender differences, and there seem to be differences associated with our body characteristics, height, weight, and age. So, if you see that European funded project, there is a chance that students of our university could perform short visit either to that artificial gravity infrastructure or to other living lab activities and have access to those infrastructures for 1 to 2 months. And they can perform, they can submit short research proposals and acquire data. Moreover, there is a compensation for the travel expenses. It is around 500 euros, I think, and there is also a daily compensation of 100 euros for each per participant for every day of the visit. Finally, I would like to present to you the methodology of a personal grant that I had during the previous 2 years. During that grant, I attempted to perform language mapping, combining behavioral and high-density neurophysiological signals. So I used 4 language experiments which were implemented in PsychoPy toolbox of Python. It is noun naming, verb naming, sentence repetition, word reading. There was also a speech recognition toolbox, the CMU Sphinx, for automatically detecting whether the participant responded correct or not. And as I mentioned, there were behavioral and neurophysiological data. We recruited 35 healthy young volunteers. Actually, the slide is a bit old. I have now also senior citizens and patients suffering from primary progressive aphasia and stroke. These are the participant characteristics. These are the language characteristics. This was the noun naming protocol. The participant was isolated during the experiment in a magnetically shielded room with 122 electrodes attached to her or his head. And we monitored from another room both the characteristics of the experiment, both the stimuli presented and also the EEG activity. This is an infrastructure that we can also exploit for high-density EEG data. Then we applied linear mixed effect model technique, which computes a random intercept and a slope for each of the variables that we attribute as a random effect. And we wish to predict a dependent variable, which in our case was the reaction time. And you may see that we reached a relatively good accuracy. This study is under publication. Again, we observed a gender effect, which was correlated with age of acquisition. Generally, female participants responded faster. I think that in every behavioral experiment, females perform better than as males, but their responses were even faster. The difference was more prominent in word stimuli that acquired earlier in our age. So there seems also to be some ways that we can improve our behavioral performance. And this way is the print exposure level. The more that we are exposed to written material, the faster that we respond to naming challenges. And again, there is a differential response between the two genders. Females seem to respond faster, especially those females that have a high print exposure. And also there seems to be a relation among how abstract a stimulus is with the print exposure level. And this again is the event-related potential, which is the averaged activity to several of those stimuli. And you may observe here the dot line is the stimulus onset. And you see here the EEG time signals and the activations on the head. Here is the stimulus, and you start seeing how strong the activations are. However, there are several difficulties regarding the spatial resolution of EEG, which is relatively low. So this is why we transit to cortical activations, and you may see again from the same participant, the same signal, how localized the activations are now on the cortical level. At that point is where the participant is able to articulate a word, and you may observe some language-specific activations here. So, last but not least, I think we already discussed it. It is a European-funded project that is currently running at my laboratory in Greece. It is the URBANOME Project Consortium, which is trying to relate climate change and air quality issues with public health. We are focusing on aging population who aim to tackle physical and mental health difficulties, and especially on the most vulnerable ones. Our motto is to shift from treatment to prevention and from longevity to quality of life. What we plan to do is to have some participatory design and citizen science activities in order to discuss what is important for each participant or for group for participants. For example, people that live close to industrial areas have different problems than people that do not have access to green or to green spaces or blue parks. So we aim to co-design and co-create the URBANOME prototype with the vulnerable participants. This prototype will be able to gather air pollution data and to estimate the exposure to particulate matter pollution for each participant separately. For example, there is different air pollution exposure to air pollution for someone who lives at West Lincoln, for example, from someone who lives at the central London areas, or where we work. The workplace is also an important issue. And we are focusing on population that's suffering from sleep-related breathing disorders, chronic conditions like diabetes, or mood and anxiety disorders. We plan to apply all those interventions that mentioned previously within the context of precision medicine in order to provide an individual approach for each participant. So I would like to thank all of you very much. I am at your disposal for discussing any potential collaborations with all of you. And remember that if we do not dare to leave the ground to jump, we will never fly. Thank you very much.
Questions? Any questions from the audience?
Yeah. I'm just curious, in one of the research you said you captured using artificial gravity, right? So how that setting